So it is a pleasure to be here with all of these great mathematicians and doing very interesting work. I'm very happy to hear about your work. I'm going to talk about some mathematical modeling that's done in drug development. People have been asking me about that. So I'll explain some detail, not full detail. And then I will also talk about some mathematical modeling that I think we should be doing that we're not currently doing as part of the drug development process. If we have time, we're running a little late, I can put in some very specific to my industry, some pieces of advice. And I'll maybe talk about my career if I have time. Okay. So let me start by giving some credit to someone who inspired me, an influential high school teacher. And this is a picture of my husband, who had a PhD in mathematics and topology, and taught at a boarding high school until he passed from gastric cancer, 2012. So I've mentioned there's a little bit of, people might hear about this through the community, or I've talked to a few of you here about this at one of the sessions I talked about it. Just put it up here so you know you can talk to me about this if you have questions, you have a similar situation, or ever wanted to ask about that. Colin's a great inspiration to my work. He really supported my work in trying to find drugs. Gastric cancer doesn't have very many drugs. Colon cancer drugs sometimes work. So we didn't have a lot of time after our wedding in 2011. But we tried to make that the best time we had. And I am an optimizer. So I do constrained optimization. And when he was diagnosed, we treated it as a constrained optimization problem. The constraints kept changing, and we were saying, okay, what are the goals now? What are the things we can change? The constraints can't be changed. So very similar to the Serenity poem that Alcoholics Anonymous use, or the Buddhist mentality, you know, I don't try to change things I cannot change, but so we always optimize. Anyway, so there's some mathematical modeling that goes on in the drug industry. So I talk about pharmaceuticals and biotechnology companies. And the reason that they do so much mathematical modeling is because the Food and Drug Administration in the United States cares about it. So why do they care so much about it? Well, here's some motivation. These are rates for clinical trials, and the first bar is just all the drugs that go into a phase 1 clinical trial. This is the first time a potential drug gets tested in a human being. FDA requires all kinds of lab experiments, all kinds of animal experiments before they will ever let us give a drug, a potential new drug, to a human being. And out of all of those, the 100% that we were doing in phase 1, what percentage make it to the next stage? Well, a little over half. So almost half of them fail. They do not progress. Once we get the results from phase 1, we think, oops, that drug is no good. We should not continue. But even worse, perhaps, is after phase 2, that cuts down to about 15% of the original number of drugs we were considering. Very bad, because the farther we go, the more expensive the testing and the more people we're giving the drug to and exposing to this potential drug. And so FDA is very concerned about this. And they give talks. They show pictures like this. These are actually some of the latest pictures that we have, because it takes a while to sift through all of the data for the submissions. But so what are the obstacles? Well, a large obstacle is prediction. This is a quote from an FDA paper that we use, still use in the industry. It's the critical path. Critical path initiative. This is what FDA thinks is really important. And I've highlighted in orange that our inability to do prediction is one of the biggest obstacles. So to be able to talk about what we're predicting or not predicting very well, let me introduce some terminology. When I talk about looking at drug concentrations versus time, we use the term pharmacokinetics. And then when we talk about, so that would be drug concentration versus time. We'll have a curve. Maybe the patient takes a pill, so you see a rise as you actually take blood samples. I'll do this. We'll have some data like this, and then it goes down later. So that's time, and that's drug concentration. And I have PK to represent that. Now, we also have pharmacodynamics. And if you're a mathematician, you might think pharmacokinetics and pharmacodynamics should mean similar things. But they don't. They're very specific, that dynamics is different than kinetics. And I don't know why the choice of those terms. But so that's for concentration I drew for the PK. Have some data points there. So now if I have some different measurement, and I do a measurement like tumor volume, versus time. Then I'll have some measurements that may increase if the drug is not working, or plateau if the drug actually has some effect. So that is called PD. So then we say PK/PD. FDA wants us to model both of those types of data. And the pharmacodynamics can be a wide variety of things. It could be a blood pressure. It could be a pain score. Could be some cognitive score that we put onto a scale. So that's our terminology. And then when we're doing the modeling of either one of those or both together, we call that pharmacometrics. So there's a quantitative science. And the United States Food and Drug Administration, the FDA, has a division of pharmacometrics. It's not that old. It's about 6 years old. And now all submissions to FDA require quite a lot of quantitative analysis and pretty complex analysis. We're doing model fitting, parameter estimation. We're doing model evaluation. And then we're going to do what we call population modeling or hierarchical modeling that I'll explain a little bit. So we typically, when we give a drug, and in this case, I'll draw a PK picture for just a drug that's given intravenously, so directly injecting into a patient's veins, intravenous. Then I'm going to have drug concentration versus time, and I'm taking blood samples. So I'm directly measuring from the same place I gave the drug. So what should those samples look like? If I draw a picture here, what's the general pattern going to look like? Okay, if you drew in the air with your finger, what would it look like? Okay, goes down. Does it go up first, or does it just go down? Some people are drawing different pictures.
So 0 is when you inject it?
Yes, and let's say I have a measurement 5 minutes later. And it'll be a concentration. So I'll take a little bit of blood, and I'll just look at how much drug is in there compared to the volume I'm looking at. Yeah, okay, so it just goes down because we are talking about intravenous concentrations. If we give a pill to a patient, it takes a while to get into the bloodstream. There's some absorption and disposition for the tissues, so that's more complicated. But I would have data, and if I draw this accurately on the scale, it'll be exponential decay probably. We have removal at a rate proportional to the amount there. Okay, so a single dose IV bolus, we call it. That's a single shot where we think the time during administration is very short. And I'm going to say that this is actually now going to be a picture of the log of concentration. And so the log of concentration will have what kind of a picture? Straight line. So that exponential that we saw before now becomes a straight line. And that hopefully, well, our data hopefully fit a straight line. And we look at that. Put this back up. Okay. So it may be one straight line. It could be 2 compartments, and that's more typical. We can have a 1-compartment drug. We can have a 2-compartment drug or a 3-compartment drug. What's happening is that I have a central compartment. This is the venous system. And we're injecting, a little syringe goes into there. This is a screenshot from a piece of software we use in the industry. And then drug may travel to another tissue, a peripheral compartment, and then travel back. We have differential equations we use to model that. And then the data, these blue dots, I think I got these blue dots from some real data. And then I fit a curve, and I've used a 2-compartment model to do the best fit possible. If I had tried with a 1-compartment, you can see I would have had to kind of hedge, and I'll miss the corner by going a little above it, and then I'll miss the end because I was a little too high in the middle. So that would be a 2-compartment model we talk about. And the 2 compartments are these 2 inside the body or something going on. And this is a bit heuristic. I'm not saying what organ, what tissue, any of that, but I'm saying I see that in the data. I know it has gone somewhere else, and it has come back, and then it's exiting the body. It's clearing there. So those are the type of data that we might look at for a single individual. This is pretty rich sampling. Might be for a phase 1 clinical trial. But now what we really do is we have multiple subjects. And this is simulated data. So I have a simulated data set here. And you can tell, perhaps, because that looks really clean, a little suspiciously clean. So I've got 6 individuals, and I've got curves fit to each of those individuals separately. But the setting we sometimes have in the later stages of clinical trials, we don't have enough data from everyone. So I'll omit some of those points. I'm missing some data. We have what we call sparse data scenarios. But I can leverage the data I have from every other subject. So I might have these 6 plus one other subject who only came in 2 times, pretty early on and pretty late. If I just have 2 time points from that 7th subject, there's no way I can get 2 slopes. No way. Unless I leverage everybody else. And that's what population modeling does. So population modeling says, not only am I going to get everybody's best fit, but I will come up with a population fit, leveraging the information from everyone else. I'll show a structure for doing that. But essentially, you can think we're putting one curve, but it's not the same as taking an average at every time point. We're tracking who's who because we expect correlation between those data points. So the correlation between those data points will have into it some variability where we're bouncing. This purple triangle bounces up and then goes back down. So you see that little bit of noise in there in the measurement or in the assay that was used to come up with the numbers I'm plotting. And so all of that's inside of that within subject variability. But then some individuals are just going to be higher than others or start high and go low. So there's some differences between subjects. We have a between subject variability structure as well. And formally, this is what we mean by population modeling or hierarchical modeling for population pharmacokinetics. This also works for pharmacodynamics. And I've now just given you equations for a single compartment. Suppose there's no peripheral compartment, just one compartment, just exit. So my differential equation's pretty simple. I have a closed form solution that's pretty simple, exponential of some rate constant times time in there. And these parameters, clearance and volume, the parameters I'm trying to estimate, I will say that they are equal to a typical value times a little fudge factor. Do I multiply the typical value by something a little more than 1 or a little less than 1? Everybody gets a little fudge from the typical value. So that typical value, I showed that one curve comes from just taking, get rid of the fudge factors for everybody. But in a population model, we estimate these parameters for the population, plus we estimate the fudge factor for every individual. And we expect some structure to those fudge factors. We expect a certain type of behavior with the distributions. And then there's a term for the residual error. Now, these are my parameters in here, but I've written an equation assuming my predictions are perfect. Perfect prediction says, here's the equation. A little bit of residual error says, your observed concentrations, C obs, are actually formed from perfect prediction times 1 plus or minus a little bit. So I have another structure for that within subject variability. That's the epsilon. Between subject variability are these fudge factors, the etas. OK, that's a little taste. I'm actually going to skip this. But there's a picture we can talk about for how those structures, what the distributions are for the etas, what the distributions are for the epsilons, and how they come together to give you a population curve, an individual, and then even the individual's perfect prediction, we're still off. We have to fudge a little bit there. So that's how those all fit together. Now, I've shown you for one compartment, IV, bolus. But typically, our drugs are 2 compartments. We have 3-compartment drugs. And we're not giving them by IV bolus all the time. We're administering them extravascularly. And there are different routes of administration. We can give the patient a pill. We can inject subcutaneously. We can inject intramuscularly. And there's some others. We have patches. Or we can do an IV infusion that's an extended length. So those go into the equations and complicate our equations. We can have that between subject variability, those fudge factors, those etas, on all the parameters in these complicated models. Usually not. So we'll have to figure out which ones have those have those variability and which ones don't. And then in a model, on top of that, we'll then go and select some covariates, some information, usually demographic information we have about a patient. So it might be their age. And we're going to see, does the clearance from the patient seem to correlate with age? Is there a pattern? Do we put that into our model? Is it a linear relationship? Is it a curve or a power function relationship? So we have different models for that we'll test. And it might be their disease state or their smoking status or something like that. We would also have lots of things we can put in the model. We can put genetics. People are putting genetics in those models and saying, OK, there's a difference. We have a polymorphism in our population. There are 2 distributions of fudge factors for clearance. Put this group over here, this group over there. They've got different structures, and we'll put that in our model. So you might imagine model fitting for these very nonlinear systems. large data sets. We're taking huge data sets and fitting very complicated models. And that can take hours, common, very common. It can take days, also common. It can take weeks. And so we're working to parallelize all of that. People are working on the software, trying to figure out best ways to parallelize. When we do this best fit, when we fit, what are we doing? We are actually maximizing likelihood. So there's a computation, and in many of these cases, our algorithms are approximating a likelihood, or they're optimizing something that approximates a likelihood. So, and actually, we talk about minimizing -2 times the logarithm of likelihood. That's equivalent to maximizing likelihood, but it's computationally there's some advantages. So you turn multiplication into addition, much better. And we are using algorithms that Livermore Labs has come up with. Alan Hindmarsh and then Linda Petzold, who came up with this automated stiff, non-stiff differential equation detector to switch. So we're using those. We're using often first-order methods. There are other techniques that have been started to be used in the pharma industry recently. These have been out there. Other industries are using these. So this is an example where algorithms or quantitative techniques have been brought in, developed in other fields and brought in. And you can see this sampling method where I do completely random selection. And in this case, I am not doing a first-order. This is a good picture I can draw. My -2 log likelihood surface. If I have a volume and a clearance in the parameters, and I'm trying to minimize my -2 log likelihood surface, it may be a surface here sitting above a plane of the parameter space. If I just had 2 parameters, I can draw it. And then I'm looking for the absolute minimum, and I'll make it a little bit more pronounced, that one of these little valleys I've drawn here for the surface above that plane is the actual minimum. Now, if I start with an initial condition over here and I use first-order methods, I may end up with something that lands in a valley that's not the minimum. And so because there is no such thing as a global optimizer, we do some hybrid approaches. We will vary the initial estimates. But that's a first-order approximation method, first-order conditional usually. There are other methods such as sampling. And that's where I say, I'm not going to try to go along the surface, which I believe is pretty smooth. Instead, I'm going to do some sampling in the space, and I'll restrict. I'll put some boundaries on the values for clearance and volume. And at each of those points, I just calculate the -2 LL. Calculating is not hard. Minimizing, very hard. Minimizing in this space. But calculating, I can get a bunch of points and kind of get a cloud picture for the surface. And I'm looking there for that minimum. That's what the goal is. And so, well, there's some smart ways to sample. And these expectation maximization techniques, the first one I'm showing is Monte Carlo. So completely at random. These I've rescaled the parameters, the space. Completely at random, you see there are gaps. You see there are clusters and gaps. That's what happens when we select numbers at random. On the right-hand side, there's a quasi-random technique. And what this does is it takes, and this is a really nice technique that was developed, where instead of doing this complete randomness, we start with something that's very ordered, and then we introduce some randomness to it by switching. If you express a number in decimals, and then you have rules for when you switch, when you randomly change one of the digits, then you'll go through and progressively, as you get in smaller and smaller boxes, as you go through the digits, you can switch things around. And you've introduced some randomness to the regular numbers that you started with. So it has good properties because you're getting some of the speed of you get the convergence properties. The errors are small because we are having low discrepancy, we call this, low gaps. But you also have speed because of the regularity properties. So it's got regular and irregular properties that have been used to prove speed and size of the error. So there's some techniques that are being developed. There's also stochastic approximation for EM methods. But that's an area of research. That's a good area of research in our field. We're very interested in good algorithms for solving. We've come a long way in the 40 years we've been doing this kind of population modeling, or 30 years maybe. But there's a lot of work to be done there. Then once we have a model, we think it's the best. Then we need to evaluate that. What do we mean by best? We could say, draw a cloud of points, draw the curve through it, and say, and eyeball it and say, looks pretty good, right? Right. Well, we really want to quantify that instead. So there are a number of diagnostics. I've just shown a few here. I might look at a predicted value, individual predicted value, versus the data. And if the model were perfect, how would this upper left picture look different? What would be different about the little dots, the circles I've drawn there? They'd be right on the line. The centers of those circles would be right on that line. So that would be the perfect model. We're looking for something that's pretty close. And that's considered a good fit, that picture. If you work with a lot of these data, you'll see bad fits, good fits, and this is one of the good ones. On the other one, I've got the residuals, conditional weighted residuals. There's a theory about which types of things we should look at. If we have chosen the correct error structure within subject error structure, then these should form a normal distribution. If that's the case, then this 2 and minus 2 are 2 standard deviations showing. And so we should have 95% of those little residuals inside of there. We'll look at trend curves, lowest curves, different ways to evaluate. We also look at information criteria, ways to evaluate. Minus 2LL, that's just a goodness of fit, raw goodness of fit. But an Akaike information criterion or a Bayesian information criterion will take into account if we use a more complex model. We're comparing models, and one has more parameters. So model 1 has some fit. Model 2 has some other fit. Model 2 has more parameters. Which model do you think just has the best raw fit? Model 1 or Model 2 with more parameters? Model 2. It's going to have, has a better fit. Of course it does. It cheated. Had extra parameters. So this is saying, no. We're going to penalize you for those extra parameters. If you want to cheat, you better be good. That's all. And AIC says, you better be pretty good. BIC says, yeah, I like cheaters even less. You better be even better. So that's just penalties. How big are the penalties? But there are many different criteria. People are working on good methods to evaluate model fits. So there's an active area of research. There are some other plots people think we should look at. I'm going to skip the covariate selection. How should we best do covariate selection? We do a forward, backward. We add in one at a time. And then we get the full model. And then we subtract, go backward. There's some literature that now suggests we should start with a full model. Some statistical modeling and trying to decide what a best algorithm is very important. And then these internal data methods for evaluating a model, we'll do a bootstrap and see how well these intervals, these confidence intervals, match our original estimate in the model. We'll do a predictive check. I'll simulate some data based on the original data set. So that's an active area of research, trying to come up with good methods to really say yes or no. We've got a model that we can use for predicting the next patient who comes in the door, or when we have 5,000 patients or more. Okay, so that is some of what we currently do. There's a lot that we don't. Well, that's being moved into that area. A couple of things I can just mention the words. There's research being done on adaptive clinical trials. So that's a good area of research. A lot of statisticians or theoreticians are working on that. If we do an adaptive clinical trial, we look at the data partway through the trial. Do we change what we're doing? If we know this group with drug A is actually not responding, do we then continue to put people in that group? So there's some techniques. And how do you no longer put people in that group but still don't lose power for your study? There are ways to do that, and that's all theory. And it's part of the work in being able to do adaptive clinical trials is convincing the clinicians and FDA that you don't lose power. So being able to explain why this is still adequate for the goals, but it's better for the people in the study. That's important. Also, another area of active research is figuring out what are the best time points to put in our clinical study. So we do what we call optimal design. And we've got variance on all our parameters that we're trying to estimate. And you might minimize the product of the variances, or the absolute value of the product. So the determinant of the Fisher information matrix is what you might maximize. It's equivalent. Or you might maximize the trace of the Fisher information matrix. So there's some work being done there. What's the best optimal design for a study? OK, so now some more work that's being worked on, not yet used. And we can optimize regimens in a different way. Basic steps, we get a mathematical model for the disease. And we also quantify the goals. What do we want? When we dose the patient, what do we want? I'll show an example. And then we will apply control theory to predict before we put it in patients. I'm going to predict before we do a bunch of tests on different drug levels or regimens. And I want to acknowledge somebody who inspired me in this field and probably is responsible for my career as an applied mathematician and getting into industry, and that's Suzanne Lanhart. Because I saw a talk she gave at a conference about HIV optimal control. I'm going to show you a paper I wrote later about a similar topic. And you know, my jaw's down here like, you can do what with differential equations? Okay, that looks interesting. And when the time came along, and I met a group at Stanford in medical school who needed some quantitative work, and I knew about the HIV modeling. And I said, oh, we could do that for this leukemia. And they said, yes, that's what we want to do. So that was the key into this new career I have. But here is, I won't go through this at detail, I'll give some references, a model for HIV where we've got healthy T cells, immune system cells that the virus attacks. And then we have infected T cells, wild type, resistant to one class of drugs, resistant to another, resistant to both. The equations that go with that model. And treatments that we might put in, I've got a protease inhibitor, so it's acting in certain ways, and a reverse transcriptase inhibitor acting in different ways. And the goal here is to optimize those. Actually, we want to quantify that goal. And I've written down an objective functional that says we're going to have T cells over the entire time period. We care about what the T cell counts are. And we also care about what the levels of these others. I'll weight them accordingly to how much I care. I really care about the resistant ones, big weight. And then these are weights on the drug amounts. It's a simple nonlinear. We know there's a nonlinear relationship. So I picked a simple nonlinear model, quadratic. So if I've written down this J functional as my goal, am I talking about maximizing or minimizing J? Who do we favor, T cells or infected viral? T cells. So that's with one sign, the other's with another sign. So which one am I going to do? Maximize. Okay, good. Because I want T cells to be high, so positive. And when this whole expression is high, that means I've subtracted off a lot. Big difference. Good. So I want to maximize. So I have a functional. I want to maximize it. So what do? Take the derivative, set it equal to 0. Very good. All right. Now we're maximizing, so we take the derivative and set it equal to 0, just like we do in calculus. But this is calculus of variations. I've got a functional, which just means I've got a function of 2 variables, 2 or more. It's a function of functions. Actually, these are functions. Sorry, I should say it's a function of functions, so it's a functional. And in fact, I've got 2 of them here. So now, just as I'll come back to that. This is the only slide I have about this. Just to demonstrate, those of you who are working in this field, you know how hard it is to get parameter estimates. I'm not optimizing. I'm not fitting. I am coming up with fixed values, although I explore a range. But these are hard to get your hands on and to feel confidence in. So we might explore a large range, and that's hard for the software. Just a note as we go by. But here are the results. And on the top, what I'm showing is, side by side, I've got comparisons between the optimal therapy predicted from the model and a traditional constant therapy. Patient takes the same level of drug every day, in fact, multiple times per day, so that when I look at a course of almost a year, it looks like a straight line. There are little ups and downs, but I can model that with a straight line. It's such a long time period. And I've set this so that the total amount of drug, the integral under the curve, or area under the curve, or AUC, as our medical colleagues like to call it, AUC, is the same in both. So the patient's actually getting the same amount of drug. No difference. Both drugs here. But the results, when I show you, I'm just showing the pictures for the T cells and the wild type infected T cells. Not resistant, but the other ones are controlled even better. So you see the 2 differences. The black curve, these are the T cells. Black is the good guys, purple is the bad guys. And you can see purple's controlled either way. However, the good guys, they dip down from the treatment, or from the system. Actually, the system's driving it to go down for a while before it can recover in those drugs. And the one on the left with the optimal therapy stays higher than the one on the right by about 100 cells per microliter. And that's considered a significant difference on a scale of 1,200. So the other key feature is that this patient, this hypothetical patient, dips below 200, which is a clinical definition of AIDS. We really want to avoid hitting that level. At that point, salvage therapies kick in. We abandon the original constant therapy. We give them salvage therapy, very harsh, very, very tough side effects. So we don't do that. Yeah.
What about that
Yeah, so this is a little bit of a viral quantity in here, and we actually, the problem from the virus is not the levels of virus, as it turns out. For HIV, you know, you can have a patient with high levels of virus, and that's not causing problems. What's causing problems is the low T cell counts. So we weight that a lot, that the T cells are the things that we care about at the end. Even if the virus is a little high at the end, the only concern I have about being high at the end is that it'll grow later and affect the T cells more. But anything it does in the middle, I'm OK with. So I'll allow it to have blips, because I'm not able to drive it to zero. If we could, we would care a lot more about that. OK, so that's not being done, but there's some work starting. We want to be thinking about that. I've got some references, and I'll put these on our Basecamp. For the first example I showed, where we've got population pharmacokinetics and areas of research, but what is the FDA guideline? A great book, Marie Davidian and David Giltenham. Marie Davidian at NC State, at Harvard for years, and David Giltenham at Genentech for years, wrote this book on this type of modeling. And so it's a great book. I recommend that. And then here are the guidelines. For that HIV, I've got a reference here, but I'll also point out I also used some of the population modeling techniques here, and we expanded on them. I don't have time to talk about it, but some recent work I had done. I've been working in cancer a couple of different projects over the last couple of years, and the one that builds on the population modeling I'll reference here. If you want to explore that a little bit more, what if you went beyond the current techniques? So what about using some cross-validation to evaluate the model fits, deciding between models? Is there a separate cell population with a separate rate? Are there two compartments or one compartment in this particular setting where we're looking at different rates of cell turnover? And so how do you quantify, yes or no, they're stem cells? They're cells that seem to have a longer lifespan. Okay, I'll take a minute or two. Yeah, the remarks are not going to take as long as told. So quick summary. I talked about this earlier. I got a PhD in pure math because it was beautiful, and I loved it, and they paid me to do it. Isn't that great? So differential geometry, tenure track at Bowdoin, working on differential geometry, went on sabbatical at Stanford, geometric analysis, same area. And then after I got my NSF grant, I actually switched areas, started working on leukemia modeling with some people in the medical school there, and I was able to use the grant money to do in the new area. And while I was in the area, started doing more disease modeling. Drug regimen optimization. Was fortunate to get a job at Genentech in the boom years when they would hire people who were a bit alternative in their backgrounds, and then move to Farsight. So that's a bit of the career summary. To mention a few things I'd like you to think about, if you're interested in this particular area, I will take the time for that slide. We're using differential equations all the time. Compartmental modeling, often. Even when I look at a diabetes model that has 14 compartments. Or I'm looking at a enterohepatic recirculation model with 7 compartments. It's compartmental modeling. We're looking at physiologically based PK models, and they may have hundreds of compartments. Very different issues, so we're doing different things with those. Sometimes with the small models we're fitting, with the large models we're simulating, only simulating. Or we're fixing pieces and fitting, and then doing the simulation with those fitted values. So I see data, and I see mathematical equations as being connected only when we have statistics. We need statistics to be able to have those connected. So that's pretty important. If I had to go back again and knew what I was going to be doing now, I would actually take some statistics instead of teaching it to myself. Software, here are some of the free packages. Again, I'll post this on Basecamp. Free packages that you can learn to do population modeling. My company has some proprietary software that maybe is easier to use, but if you know these, that's great. In many of your programs where you are currently, if you're a grad student, postdoc, you can go over to pharmacology or medicine and take a class. And that would be a good intro. If they have a pharmacokinetics class, great intro. If you wanted to try to do a postdoctoral program, here's a list that I'll post of programs that are very quantitative and have great success with their students coming out of those. And I mentioned some of this before, but when you're looking at company names, here are some modeling companies I was able to think of: Entelos, Rosa, Merrimack, and Silver Creek. These last two are actually drug development companies. They've got molecules, but they're based on modeling. It's all modeling, very complex modeling. Entelos and Rosa are more just, we do modeling for any company. But that's the list of those that have have that focus if you're looking for some that have more of a focus there. And that's it. Thank you for your attention.